mRNA Electroporation provides a fast, efficient, and spatially and temporally controlled expression of multiple proteins within the Avian model system. This method allows for a more broad and quick expression of fluorescent proteins than the labeling achieved by standard DNA electroporation.
Electroporation facilitates the transient opening of pores in the plasma membrane that serve as conduits for the transfer of genetic payloads like RNA and DNA into the cells of numerous model organisms. For the initial experiments, work with embryos that are older than a day old, since they are hardier and more resistant to external manipulations like electroporation.
At the appropriate embryonic developmental stage, gently break and pour the quail egg contents into a 10 cm petri dish. Use a transfer pipette to remove the majority of the thick albumin. Use a lab tissue to gently wipe the surface of the yolk to remove the remaining thick albumin around the embryo to ensure that the embryo will stick tightly to the paper ring. Lay pre-cut filter paper over the embryo and use scissors to smoothly cut around the perimeter of the embryo. Use a Pasteur pipette to layer PBS under the embryo, using gentle streams to vacate any yolk sticking to the tissue. Slowly pull the embryo paper ring at an oblique angle up and off of the yolk. Place the paper into a petri dish filled with PBS for further cleaning. Once most of the